All right. I'm a little sad now because I think this is we're down to the last talk that's not from ORS personnel and ORS teams. So, but I'm sure we're going to make it a great one. So, Doug, I don't have my note card in front of me. How long have you been using Dragonfly?
Since the pandemic, I would say, but I've known about it longer.
All right, a pandemic convert. So Doug is faculty at UC Davis, so principal research scientist at UC Davis, and he's going to tell us about in vivo and specimen imaging at the Center for Molecular and Genomic Imaging. Are you going to see the sample I asked about? Are we going to get to see the COVID mice?
Yes.
Oh, good. All right.
You gave
All
away
right.
my presentation.
I was. Oh no! All right. Now that I've ruined it, we can all just go to. No, let's turn it over. Turn it over to Doug. Thanks.
Thanks. First, I want to thank ORS for inviting me here to give the talk today. It's a great opportunity to show what's going on at UC Davis and what we're doing with Dragonfly now. And it's amazing seeing all the presentations and how it might touch each of us. The last presentation in particular, I broke my hip a few years ago. And it makes me wonder, it was femoral head fracture. And it makes me wonder what it looks like in there. But you can't have my bone to analyze it right now. But anyway. So I'm the principal research scientist at the Center for Molecular and Genomic Imaging. I'll tell you a little bit about the center. My background is in chemistry and physics and technology, instrumentation, construction. So I will be presenting some biology. Biology I've picked up along the way. So please forgive me if I misstate anything. I'll try and present it as coherently as I can. So I transitioned into nuclear medicine imaging from my PhD to do in vivo imaging in animals. And so what we do is we have animal research models, and we want to use those as models for human disease. And the instrumentation that we use have clinical components. So we have PET, CT, MRI, and all of that instrumentation. And you can see here on the right examples of images from the human brain. I have a little bit of discordance between the instrumentation and my images. The two scanners that I show there are PET CTs. The upper one is the newest instrument. It got installed last week, so we're really happy to have new instrumentation. But the images that are shown here, the grayscale underneath is MRI anatomical imaging, and overlaid on that is PET imaging for PET is a technique for physiological imaging. You might be most familiar with it from fluorodeoxyglucose scanning to look at metabolism in the body. And so at the Center for Molecular and Genomic Imaging, we're a campus research core facility. I liked Ramon's presentation, especially when he showed the recharge rates. I don't have mine here, but we do publish those on the web. But our whole core mission is to have state-of-the-art instrumentation that not everybody can have, and allow people to come in and use that instrumentation for their research projects. And I think I have a great job because I interface with a lot of different scientists and gives me the opportunity to see a lot of amazing science. So I made this slide for a talk that I gave a few weeks ago at Carl Zeiss, and it's mainly based on micro-CT imaging that we do, but it really does encapsulate a lot of the other programs for the in vivo imaging that we do. So you can see the array of departments. We also do recharge work for academic institutions and industry as well. So again, the instrumentation that we have, I mentioned the different techniques. These are all instruments in our lab or satellite facilities. For PET imaging, the image, is there a laser pointer? pen and laser pointer tools. Let's see. So this instrument here is actually our biomedical cyclotron. So for PET imaging, you need radionuclides production, and so we have an on-site cyclotron to produce the radiopharmaceuticals for PET use. But today, like most people here, I'll be just presenting on micro-CT imaging. One example of in vivo imaging, and then another example of specimen imaging. So the two instruments that I show here, this one on the right is the old X-Radia Micro XCT 200. It's our workhorse for specimen imaging, and it provides beautiful images at high resolution. And I'll show you one of the most recent examples of that in the second half of my talk. But on the left here is one of our newer instruments. We got this in late 2021. It's a micro CT scanner that we got from a supplement to an NIH grant for COVID research. And what's special about this one is it went into an animal BSL-3 facility. COVID research has to be undertaken in a higher biosecurity level. And so we have to get dressed up in bunny suits, masks and hairnets and all kinds of PPE when we go in to study the animals. It's a little bit unusual. There's not a lot of instruments that go into BSL-3 facilities. So we think that it's quite a unique facility. And you can see that the instrument is in the animal room. These are the animal cages on either side of the room, which is also a little bit unusual to have the equipment directly there. So what I'll present about the COVID research is we want to establish an in vivo model for post-acute sequela in SARS-CoV-2 infection. This is the kind of rebranding of long COVID. So you can think of PASC as long COVID. And you may know people that have experienced that. And we want to study this in a novel, genetically humanized polygenic mouse. And these are the main investigators on that project. So the rationale for this is that we want to understand the pathophysiology and also evaluate therapeutics that could be moved forward into the clinic. And current animal models have disadvantages. You can see monkeys here. The main disadvantage of them is that they're expensive and limited in terms of the number of animals that you can utilize to study these models. Mice and hamsters have their own disadvantages, and currently what those would be is the genetic modifications that have taken place to replicate the disease in the human population. So we want to really develop the characteristics of human COVID in the animal models. So what we'd like to do is confirm the infections that we present to the animals, look at their survivability, and can we do in vivo tests. So to genetically modify the animals, the current models, they insert the genes and genetically modify them in kind of a random way. And so the genes that are important for COVID infection are these ACE2, the temperus, and furin coding sequences. And rather than just throwing the genes into the animals and having them expressed in many different cell types and in random locations in the genome, what the mouse biology program at UC Davis has attempted to do and has done is to introduce those genes in the locus where the genes exist in the mice. So using CRISPR technology, they're able to knock in the genes into the mouse-specific locations and replace the mouse gene with the humanized gene. And so far, what we've done is looked at the ACE2 and the ACE2 temperus animals. In the future, we'll be looking at the triple CRISPR knock-in mouse. So You can see here the hypothesis that female mice would develop mild to moderate COVID-like symptoms acutely and are more likely than male mice to exhibit PASC symptoms, or mice exhibiting severe sublethal acute disease also developing PASC within 6 months. So you can see the research plan here. In our AIM 1, we're just now taking that data, and we'll be evaluating AIM 2 to evaluate the PASC symptoms. So I'm not going to, so in the entirety of this experiment, there is testing for whether the animals were infected, either by measuring the virus directly through nasal swabs or looking for antibodies in the animals, and then following them long-term with various tests, either behavioral tests, micro-CT, or some metabolic tests. So what I'm presenting today, though, is only the micro-CT imaging. So what we want to do was look at respiratory-gated lung imaging. And you can see here the total number of days that we've investigated the animals. What I'll show today in some data analysis are just the first 4 time points, but we have looked at them over monthly scans for about 6 months. And you'll see that the later days here are closer together. That's because we did do a smaller number of animals where we reinfected them with COVID. So they would get exposure twice. And then we want to look at the lung injury due to the infection. So in these images, what you can see are the basic lung structure of the mice. And in this first column, you can see a single animal at baseline, and then at day 5 and day 17 post-infection. And what we observed was that in the ACE2 temperus mice, you can see that in this animal, there's a little bit of hyperintensity right where the arrow points. But at day 17, that hyperintensity is gone. So we equate this to active infection at day 5, where there's some pathophysiology in the lung. And we did observe this in, we had a total of 16 animals that were infected. And we observed this in 5 of the 16 animals. So not all of the animals exhibited it. And the arrows in these 3 animals at day 5, you can see some hyperintensity in those. So we can collect a lot of data, and I wanted an easy way to do the segmentation, because manual segmentation just would not, it would be unimaginable to try and do manual segmentation on this. So this is, I think, hopefully people are interested. This is the PC that we built to be able to process micro-CT data. I'm lucky enough this is sitting on my desk and I get to use it every day. But a lot of RAM, a great graphics card to do the deep learning. But I still want to push it more. So in the deep learning model that I'll show you today, these are the basic parameters. We used a sensor 3D model. We did use data augmentation, and you'll see, just remember these numbers here for the image mask, because I will note that in this second half of the talk. This is a little bit smaller than high resolution data on our specimen scanner. And then you can see we trained with 6 training sets, both at end expiration and end inspiration. The end expiration images have a higher signal and noise because of how the data is acquired. And so using both gave us the breadth of signal and noise in the images to use a single model. And then there is some data cleanup that I would like to mention. The model isn't perfect. There are blobs outside of the lung, but the model captures the whole lung in most cases. There are some unusual cases where it doesn't, and I have to do some cleanup. But in most cases, it's very good lung segmentation, sometimes little blobs outside of the lung which are easily separatable. So we did do some in the training and the validation. You can see in the upper left that the loss function over the number of epochs. So I did do a monitored validation loss for early stopping. And you can see that I had it set for 200 epochs, but it finished after 52. You can, as I described the two images, you can look at the table for the accuracy in the dice, but I think the model is doing very well. What's represented in the two images, the red are the false negative voxels, and the green is the false positive voxels. So while it looks not so good when, I wish I would have done a movie of this. I'm still learning Dragonfly and how to do movies. But if you look through the slices of these, all of those voxels are at the boundary of the lung. So I'm not so worried about that. The model is doing consistently animal to animal to animal. And so as long as that's the case, I think the divergence between my manual segmentation and the model is not so important. And so this is some of the early results. And so what you see is that in the SARS-CoV-2 infected animals, we see a trending towards day 5 for decreased lung volume in the end expiration images. This was in just n equal to 8 at this point. I still have to add in an additional 8 animals to the SARS-CoV-2 group, as well as analyze the rest of the time points. And so you can see that at the end of the study, I have about 264 end inspiration and expiration images, and doing 1 to 2 hours of manual segmentation really would not be feasible. So that's the end of this part, and I would like to thank the first 3 are members that help with all the experimentation. I have to thank Abhijit and Valerie because they helped me a lot in learning about deep learning. They're in the biomedical engineering department, and they were of immense help, as well as the ORS team to help me get situated in the deep learning world. So to move on to the other project, which, sorry for the divergence in the backgrounds, but this is a project of Denise Ramirez, and I must thank her very much for the slides. I've gotten her slides unaltered because I think they're very beautiful, and hopefully you'll see that her use of Dragonfly was, she's picked up on it very well. So she's looking at ontogenic signals of sexual dimorphism in a neotropical orchid bee. And so what is an orchid bee? When I think of bees, I think of honeybees and bumblebees, but there's a whole group of species of orchid bees that are a diverse group in neotropical areas. And so there's an enormous range of behavioral diversity in these bees. And so there's social species, as well as kleptoparasitic species, and bees that have scent collecting behaviors. And this is an example of a male bee that is doing its scent collecting behavior. So it goes to the orchids and other flowers and collects the scents of the flowers. And they'll put this on display for the females to attract females. And so this is an emerging model to tackle some neurobiological research questions. And so what's interesting about this species is that there is a divergence in behaviors between the females and the males. And so females are semi-social, and the males are mainly solitary, and they collect the perfume, whereas the females do not. And so this sets up a very interesting research question of how does the brain evolve to support this behavioral diversity? And so the question then is, are there signals in anatomical sexual dimorphism in the brains of these bees? So to study this, Denise came to our facility asking about micro-CT imaging and what we could do. So I would say that, so Denise has been working with us for about a year. I think this is a good example of how fast research can take place with micro-CT and with appropriate software. So these are examples of the bee brains that have been scanned. It took a little bit of work to get the correct staining procedures down. We have to thank Brian Metcher at the University of Vienna for helping us determine some of that. The stain that we used was iodine and ethanol to do the staining. And so we get very good contrast in the different brain structures in the bee brain. And so this allowed Denise to do some manual segmentation of the bee brain, which you can see here. And I had mentioned the voxel size. So even though the bee brain is much smaller than the mouse lung, it ends up that the matrix dimensions in the bee brain are about 2.5 to 3 times the matrix dimensions in the mouse lung. This has a big consequence for deep learning, which I think you can imagine. But before I get to some of the results and talk about that, this is a really nice movie that Denise made for the bee head and looking at the structures inside. And so I looked back at our communications, and Denise has been using Dragonfly since about October. I gave her a little bit of input in starting, but she was able to take this and run with it and produce all of this on her own, either through watching your videos, Mike, or going through the documentation and everything. So she did a wonderful job on this. And so what she did a manual segmentation of were 9 brain regions in the bee brain. And she'll be looking at the volumetrics and morphology of these regions. And so you can imagine 9 regions in the bee brain over the around 1,000 slices in the volume. It's a lot of manual segmentation. I think she started out that a single bee brain would take about a day to segment, and we talked last night about 2 to 4 hours is about what she's down to. And she does want to study multiple examples of these, potentially up to 100 of these. And so I think deep learning will be a very important aspect of this. And the problem that I'm having right now is finding the right tuning parameters and downsampling to get the data we need in an appropriate training time. So that's some of the things that we're toying with right now. And so the last thing to show is a little bit of what she's already found in the few samples that she's been able to manually segment. And so in the lamina and medulla of the brains, she's found enlargement in the male bees, and these areas are important for the visualization of the bees. And this is important for finding the flowers and being able to collect those scents. Whereas in the female bees, she's found that the mushroom bodies in the bees are enlarged. And this is important for associative learning and memory, which is important for the social aspects of the bees. And so there does seem to be this ontogenic signals in the anatomical dimorphism. And this is her research group that has helped to support her through the work. And she presented this work at Carl Zeiss workshop a few weeks ago, and that's why they're on this slide. And with that, I will take any questions. And these are just some images from our micro-CT that I've acquired over the years.
Well, thank you for that talk, Doug. Those the contrast on those bee images and looking at the spatial resolution, the contrast resolution of seeing the anatomy is is remarkable. And and seeing the COVID mice data where you can actually do the temporal resolution and do these respiratory gated studies and ask these physiologically relevant questions is also very impressive. In the interest of time, let's do one question now, and then let's do lunch. We are going to have until 1 p.m. For lunch, but let's take one question from the group if we have one before we dismiss. And then I have 2 announcements before we dismiss. Any questions for Doug right now? Or will you, if nothing urgent, then you can just connect with Doug during the lunch break. All right. Let's thank Doug one more time.